Hello, good afternoon, good morning, all of you. Come on. In this class, we need to talk some fundamentals of secondary growth. Hello, anybody there? Hello, good morning. Voice is okay? Okay. Shall we begin? Is everything okay? Come on. See? Up to the last class, we have discussed about dicotyledonous stem, root, leaves, and also monocot bodies. Next, come back to we will discuss some small concept that is called secondary growth. Secondary growth. Secondary growth. Let us check the details. Imagine when we talk about secondary growth, the first question will arise: is it there in NCERT book? Of course, it is not there. But we have to study. Why? The reason, this part is not directly given in NCERT book, but in different places, this concept has been explained in different orientations. That is, there is a lesson called plant growth and development. There you have seen several words which are almost similar to secondary growth or similar to secondary growth, let us take. That's the reason why we need to come across the secondary growth. How much is required, that much I can able to explain properly. Along with me, as usually you have to write the notes. Then whenever I ask something to reply, try to reply faster. That's a deal. Let us move. First one, before secondary growth, let us begin. What is the definition of secondary growth? Secondary growth indicate increase in girth of the plant. Girth of plant, that means circumference, or width of the plant, width of organs. This is called secondary growth. Now, if you want to catch exactly what is secondary growth, you need to talk about what is primary growth in first minute. Primary growth. Tell me, what is primary growth? Primary growth, it is due to increase in length or axis wise. Axis, which means plant axis, if it is growing, this is called primary growth. Now, primary growth, when we observe, it occurs due to apical meristem. Apical meristem. So sometimes what they even say, primary growth is also called apical growth. At the apical portion or apex region of the plant, this growth continuously occur. That's the reason why primary growth is also called apical growth. Now, when it comes to the word primary growth, it's a broad term. It does not define only increase in the length. It also includes formation of leaves, branches, flowers, etc. etc. Anything that will be associated in the early stages of plant growth, which is known as primary growth. Primary growth is occurred due to primary meristem or apical meristem, is exactly to say apical meristem. At the time of apical meristem discussion, I told you Apical meristem is the one which forms leaves, which are called leaf primordia. Apical meristem is the one which form epidermis, ground tissue, vascular tissues. So, apical meristem or primary meristem responsible for formation of almost all organs. That's why this apical meristem growth is universal. Universal. Every plant that existing on this earth, you take bryophyte, pteridophyte, gymnosperms, angiosperm, whoever it may be. If it is a plant, it must be having the primary growth. With that, we will have a full stop to primary growth and come back to secondary growth. Secondary growth, it is a kind of transversive growth where the plant organs increases their circumference or width. Now the question, does it occur in all plants? No. It is exclusive for dicots of angiosperms and also gymnosperms. Gymnosperms both will perform secondary growth. That too, if the question arises, do all dicots perform secondary growth? Absolutely not. Many dicotyledonous plants, which are annuals, they do not perform secondary growth. Generally, I am saying, okay? There, for example, lettuce. Lettuce is a dicotyledonous plant, but it cannot perform secondary growth. Why? The life ends within 1 year. In one growing season, they can complete their life. That's the reason why there is no need of increasing of circumference of their body. That's one thing. Next one. Do gymnosperm perform secondary growth? Yes. Do all of them? No. Wherever you find certain kind of concept, here and there exceptions should be noticeable. If the given statement, all dicots perform secondary growth, no. Dicots perform secondary growth, yes. And then come back, whatever the secondary growth you are going to be discussed, that secondary growth we will discuss only associated to dicot. Gymnosperm secondary growth we don't touch. Next, in that dicot, observe secondary growth is due to two types of meristem. One is called vascular cambium. Vascular cambium. Next one, cork cambium. These two meristems will be developed in two different areas of the plant, and they will be responsible for secondary growth or increase in girth or circumference of the plant. Let us see how a dicotyledonous plant can perform secondary growth. For my convenience, I am dividing this dicotyledonous secondary growth in two parts. One is called intrastellar secondary growth. Intrastellar secondary growth and extrastellar secondary growth. Extrastellar. I think you might be having an idea what is a stele. Stele indicate all the tissues that are present inside the endodermis, known as stele. The tissues that are present exterior to the endodermis, known as extrastellar area. Extrastellar area, one meristem responsible for the secondary growth. Intrastellar area, another meristem. Intrastellar area, vascular cambium is responsible. Extrastellar area, cork cambium is responsible. First, I will begin intrastellar secondary growth. In this, observe, I am taking a stem. Imagine it's a epidermis. Epidermis. Below the epidermis, what will be there? Hypodermis. Do you remember hypodermis made up of what type of tissue? Collenchyma. Collenchyma below that, parenchymatous ground tissue. Below that, I am talking all the tissues associated to primary meristem or primary growth. Okay. Below that, there is a presence of endodermis. Endodermis, below that, sclerenchyma. That sclerenchyma present in the form of semilunar patches. The semilunar patches of sclerenchyma is also known as pericycle. Below that, vascular bundle. Vascular bundles are what type? In the dicot stem, conjoint, collateral, open type. Then inside that there is a presence of pith. Between the two vascular bundles, medullary rays. Okay. Now I am choosing only those tissues that are required for understanding of the intrastellar secondary growth. If it is epidermis, there should be a presence of endodermis. Observe this, I am choosing it as endodermis. Now, in pericycle, general cortex, okay, you know all of them. Inside that, they have ring-like arrangement of vascular bundle. Do you remember that term? Ring-like presence of vascular bundle is a characteristic feature of angiosperms. Ring-like vascular bundle is a characteristic feature of angiosperms. In that case, the vascular bundles I am writing like this. These are vascular bundles. These are all ring-like arrangement of vascular bundles. Be careful, one vascular bundle shape is not ring. Be careful. I have seen many students getting confused. What is the shape of vascular bundle in dicots, dicot stem? That should be either oval shape you can write, or you can write it in the form of wedge shape, that is trigonal, like this shape. This is one vascular bundle shape. All the vascular bundles are arranged like a ring. This arrangement is called U-stele. Okay, well done. Then go inside one vascular bundle. We have seen that there is a presence of intravascular cambium, because these vascular bundles are conjoint open type. This is, I am choosing it as intravascular cambium. Below the intravascular cambium, there should be a presence of primary xylem. That primary xylem I am decorating like this. This is primary xylem. Peripherally, there should be a presence of primary phloem. These yellow dots, I am choosing them as primary phloem. So, then go to, this is primary phloem. This one, primary xylem. Okay. Between the primary phloem and primary xylem, there is a presence of a band of cambial tissue. The cambial tissue is known as intravascular cambium. Could you tell me what is the other name of intravascular cambium? Intravascular cambium is also known as vesicular cambium. Is it primary or secondary? Of course it is primary. That's what we have learned in the earlier classes. Okay. Whatever that thing I have written here, this is not after the secondary growth. Before secondary growth, these are the tissues present. Now let us begin. If the secondary growth is beginning intrastellarly, first step is this. During at the time, observe carefully. This area, what do you call? It's called pith. This area you call medullary rays or pith rays. Now, the pith rays cells are medullary rays cells which are present adjacent to intravascular cambium. They undergo dedifferentiation. As a result of that, they will form a cambial band. This cambial band is known as what we call interfascicular cambium. Interfascicular cambium. Interfascicular cambium. This one. One band is already present in between the primary xylem and primary phloem. One band is newly formed. From where? It is formed from dedifferentiation of medullary race cells. Medullary race cells have gone dedifferentiation. The word should be particular, perfect. That is dedifferentiation. You should never say differentiation. Differentiation is a different term. Here, meristematic cell has been formed from primary permanent cells. That's why it is called as a event called dedifferentiation. Okay, done. There is a band of intrafascicular cambium, another band of interfascicular cambium. Both combinedly form a layer that is called vascular cambium ring. Vascular cambium ring. Okay. Does this vascular cambium ring completely secondary or completely primary? It is partly primary, partly secondary. Which part is primary? Partly primary, I am saying. The part is primary is intrafascicular cambium, and it is partly secondary in origin because interfascicular cambium is secondary. Okay, successfully there has been formation of a vascular cambium ring. Some part is primary, some part is secondary. Okay, vascular cambium ring has been formed. Formation of vascular cambium will allow these cells. Cambium means what? Meristem. These cells can able to divide. The cells which are dividing from the cambium started to give new layers of cells. What are they? Observe this. Imagine. In this, carefully follow me. This is vascular cambium ring. Inter, intra, inter, intra. Alternatively, intervascular cambium has formed vascular cambium ring. This vascular cambium ring started to divide. These divisions we are called cuttings. What do you call them? Cuttings. The meaning of the word cuttings means they are dividing. Cuts, division, almost same. Anyhow, they have some differences are there, but the difference is not necessary for us. But here remember, cuts means divisions. They are dividing. Vascular cambium is dividing. It is dividing. Towards the inner side, it forms one variety of layer. Towards the outer side, it will form another layer of cells. One variety of tissue will be formed towards the inner side. Another layer of tissue will form towards outer side. In this case, now let me confirm what is happening in this part. Careful. The vascular cambium started to divide. Observe, this is that vascular cambium, what you say. This vascular cambium towards the inner side, it form secondary xylem. Towards the outer side, it form secondary phloem. Towards the outer part, secondary. Towards the inner part, secondary xylem. Towards the outer part, secondary phloem. In this case, one thing you have to remember, vascular cambium grow more growth towards the inner side when compared to outer. Outer will grow less. which means secondary xylem quantity several folds excess when compared to secondary phloem. The amount of secondary xylem formation would be more when compared to the amount of secondary phloem formation. That's why we say the activity of vascular cambium more towards inner side when compared to outer side, which means secondary phloem formed less in quantity when compared to secondary xylem. This is the onset of secondary growth. Secondary growth happened, it is continued. Sometimes we say the divisions of vascular cambium is cuttings. You will read the statement like this. Okay, this vascular cambium cuts off and produce secondary xylem towards the inner side. Secondary phloem will be produced towards outer side. Cuttings nothing but divisions. There is a variation is there, division and cutting. Observe, if a parental cell divided and given two daughter cells, both are similar to parental cell, this is commonly denoted as division. A parental cell divide and given some cells, but they are nowhere related to parental cell. This is called cuttings. Cutting, division, okay. But why did they say here cutting? Cambial cell is meristematic. This cell has given xylem, 4 different types of cells which are nowhere related to meristem. Outside 4 types of cells it has given, they are also nowhere related to meristem. That's why these divisions are called cuttings. Okay, division started growing, let it grow for a while. After few years you will see, be careful, secondary growth does not occur day by day, gradual change. It occur year by year, somewhat slowly it occur. Okay, so after few years we are going to take this secondary xylem and secondary phloem formation. That one I am choosing here. There, look at the secondary xylem and secondary phloem. Observe, this is the secondary xylem. Secondary xylem has been formed. This secondary xylem has occupied most of the area of our okay, stem. This is that. Outside it, who will be present? There will be a presence of vascular cambium ring. Vascular cambium ring is present. This is that. Vascular cambium. Outside the vascular cambium, who will be present? There will be a presence of primary phloem. Okay. You follow slowly and somewhat patiently. Don't make it hurry. Then these are the layers. See, nothing I have drawn here. Whatever the tissues that you have taken here, the same tissues after few years, how they will be changed. Those changes I am explaining. That change is this. This is our vascular cambium ring. Vascular cambium ring. This is secondary phloem. This one is secondary xylem. Okay. Observe. Secondary xylem developing gradually and it is making inner side area slowly get decrease. Could you notice that? it is making the inner area slowly get disappear. But actually, the inner side area, this is secondary xylem. Inside it, who is there? Primary xylem. That primary xylem fate we have to decide. You can notice outside the secondary phloem, there is a presence of primary phloem. What is the fate of primary phloem and secondary phloem? We have to decide now. That's why I am writing that diagram. See, after few years of secondary growth, now primary xylem, this is primary xylem, more or less intact. Even though it has been crushed inward, the primary xylem more or less intact, but primary phloem disappears. Primary phloem will get disappear, gradually crushed away and disappear. Who will get disappear? Primary phloem. Imagine this is primary phloem and this one is primary xylem. First we need to confirm what happened to them and then we will go to further. Primary phloem gradually crush off. Its work will be gone. There is no existence of primary phloem. And you could see inside, I have written primary xylem. The fate of the primary xylem in initial days, more or less intact. It will be there, but maybe it will be there as it is, or it may be there due to some changes that varies from plant to plant, but it would be there. That's what we need to learn. More or less primary xylem will be intact. I am not saying primary xylem forever will be intact. Primary xylem would be present there in initial days. That means after few years, you can find the growth continuous, but still primary xylem will be present. But primary phloem would be crushed away even in the initial years of secondary growth. Got it? This is about the fate of primary xylem and primary phloem. After few years, what will happen? This entire area completely filled by secondary xylem. There is no further existence of primary xylem over there. One more time. If they say after beginning of the secondary growth, primary xylem more or less intact, correct. But if they say primary xylem would be present forever inside the secondary xylem development inside a stem, it would be absolutely wrong, because secondary xylem while growing, initially it will crush away primary phloem, but later gradually it will crush away primary phloem, primary xylem also. It doesn't mean that the primary xylem could be present forever. Understand? Then come back. Next one. In this case, after few years, what are the changes that you would notice in the secondary growth of a dicotyledonous stem? That thing we will understand before that. Secondary xylem has another name called wood. Secondary phloem has another name called bast. If you recall, these words I have already given to you. Secondary xylem is known as wood. Secondary phloem is known as bast. Okay. Next one. Let us observe about, carefully listen, about what happened after few years. To understand that, we need to know one thing. Vascular cambium activity depends on physiology and environment. Depend on what? Physiology and also environment. Environmental conditions will affect vascular cambium growth and also physiology. Physiology means internal actions. Imagine a statement, a plant got diseased. In a diseased plant, you cannot expect the vascular cambium to grow actively, or environmental conditions become change. You know, there are seasons which make the plant to change its different types of internal mechanisms. You cannot expect a plant to behave identical for all seasons. So environment will affect the vascular cambium growth, and also vascular cambium growth would be affected by physiology of a plant. First I am talking with respect to environment. For that, let us consider there are some plants called temperate zone plant. Temperate zone plant. Do you know what is temperate zone? See, tropical zone, temperate zone, both zones if you understand, just only two zones understand. Temperate zones are nothing but those areas climatic conditions are very sharp and mostly the year covered by snow. That means cold climatic conditions are called temperate zone. But we are living in subtropical zone. It's not completely tropical, it is not completely cold, but we will have subtropical climate, often denoted as tropical climate. You know what is the tropical climate? That's a hot climate. Next, observe, when it comes to temperate zones, in the temperate zones the story started, they have sharp seasonal changes. Seasonal changes. Sharp seasonal changes. What does it mean by sharp seasonal changes? One season to another season, the environment totally changes. Let us see in our India, we will have only one season throughout the year, summer. Sometimes a cyclone come, we will feel rainy season. During the December and January month, during night time, some amount of temperature will drop, then we will say it is winter for us. Such kind of things happen in our India because we are living in subtropical climate where seasonal changes are not very much sharp. But take some countries where snowfall will occur in winter. In these countries, the seasonal changes are very sharp. What is the seasonal changes? In winter, snowfall begins, soil covered by snow, plant do not get water and they could not grow. Most of the soil surface covered by ice, so plant and grass cannot grow. Then ultimately the plant growth will be controlled by snow. If plant growth controlled, automatically animals could not grow there. The entire population of plants and animals come decrease during winter. Next, winter may last for few months. In fact, it lasts for many months. In case of temperate zones, around 4 to 5 months, winter will be continue. After the winter, temperatures gradually increase. The increasing of the temperature will make summer. Summer is the best season for their growth. Climatic conditions become little bit warmer and then the plants take water from the soil and they can expose to sun and they can survive. This is the thing why I am going to confirm with one statement. Summer is called as growing season in case of temperate zones. Summer is the growing season. Before the summer, winter. After the summer is also winter. But if winter going away, gradually temperatures will rise. The temperatures rising season is known as spring. Summer, plants are growing. Again after the summer, temperatures started to drop. The dropping of temperatures is known as autumn. Simple. After summer, autumn will come. Before summer, spring will come. Spring is the early to growing season. Autumn is late to That's why when you talk about spring season in temperate zone, it is called early season to grow. What is actual season to grow? Summer. Before summer, spring. That is early to growth. After the summer, autumn. That is late season to grow. Very good. In this criteria, in temperate zones, because of the sharp seasonal changes, vascular cambium activity also changes. Vascular cambium activity also changes. How? First, I will talk about during spring season. In the spring, where the temperature started to rise, okay, vascular cambium becomes more active. As a result, slowly understand. During spring, vascular cambium become more active. As a result of this more activity, xylem elements are nothing but xylem vessels produced with wider lumen. They are produced with wider lumen. If the wider the lumen, more the water can they transport so that the plant can grow healthy. That's the intention. As a result, during spring season, whatever the secondary xylem will produce, secondary xylem is nothing but wood. That would appear in lighter color. Lighter color. That lighter color wood is known as early wood. Early wood, or it is also called spring wood. Come on. Spring wood will be in light color or dark color? It is in light color because the lumens have been formed are wider. Next, during autumn. In autumn season, vascular cambium becomes less active because temperature started to fall. Okay, it becoming less active. As a result of that, vessel lumen become thick, our cell walls become thick, vessel lumens become narrow due to the formation of thick walls. Thick walls will be formed so that the vessel internal luminal volume will be narrower because cell walls are going to become more thick. Due to the presence of thick walls, the wood which is formed here, it become dark color wood. This dark color wood, it has a name called late wood. Why it is called a late wood? It has produced late in the late wood would be also called it as autumn wood because it is produced in autumn season. Like this, if you see those plants which are growing in temperate zones, in their secondary xylem, one band will be dark in color, another band will be light in color. The dark color band is known as autumn wood. The light color band is known as spring wood. You can see there what I have written. The outermost is the dark color band. The dark color band indicate autumn wood. This is the light color band. The light color band indicate spring wood. A band of a spring wood and a band of autumn wood collectively known as annual ring. An annual ring. Annual ring. Remember, in 1 year, one spring season, one autumn season. During the spring season, light color wood. During autumn season, dark color wood. Alternatively, a band of dark wood, a band of light would be developed. This alternative band of woods could be helping to measure the age of the plant. So one dark color band, one light color band is known as annual ring. You can measure age of the plant by measuring annual rings. By measuring annual rings. This mode of measuring of age of the plant is known as dendrochronology. Dendrochronology. Dendros means, come on, dendro means, do you have any idea what is the meaning of Latin word dendro? Dendro means tree. Do you remember? You have seen there is a word called dendrites. If you have brain, in the brain there are neurons. Neurons will have axon, dendrite. Dendrite appear like a tree. That's why they are called dendrites, tree like bodies. Next one, chrono. Chrono means, can you say what is chrono? Chrono means time. Chronograph means watch. By using annual rings, we can approximately measure age of the tree. This technology is known as dendrochronology. Tree age or dating of tree is possible by the annual rings it will develop. This technique is known as dendrochronology. What do you call that? Dendrochronology. Observe, the dendrochronology technique will not be applied in our India. Why dendrochronology could not be applied in our India? Simple. If you go to our neem plant, which is a temperate zone plant, which is a tropical plant, that tropical plant will never expose to any kind of seasonal changes. Throughout its life, it will never see cold or snow environment. So it doesn't have a chance to develop any kind of variations in their annual rings. Is it clear? That's why dendrochronology not useful in Indian plants. In India, this will not work out. But in our India, some places, there is some area called Himalayan jungles, and also in Jammu and Kashmir, some places where snow falling is possible. There, this dendrochronology may be work out. But in our especially South Indian climatic conditions, dendrochronology does not going to work. Okay, this is one methodology. Next one, based on physiological conditions. You said environmental, then you have taken the plants which are living in temperate zone. The temperate zone plants, some year they will in an year some part they will grow better, some plant they will some a part of the year they cannot grow. So ultimately the temperate zones will produce annual rings. Next one, based on physiology. There I am talking based on maturation. Based on maturation character, if you are talking, then you will find in some plants the vascular cambium, that means secondary xylem in the center area, it become too much dark. In some plants, this could be seen in both tropical climate as well as subtropical climate, and in the center the wood is going to become very dark in color, but peripherally the wood become light in color. You can understand, this is peripherally light color wood and the centrally dark color wood is present. This is this. Over there, you notice one thing, This centrally located dark color wood, it commonly known as dark color wood, is known as heartwood. Heartwood. Peripherally located light color wood is known as sapwood. Sapwood. Now question will come. Why this heartwood or hardwood is dark in color? It is dark in color due to deposition of tannins, resins, and also oils, essential oils, and some variety of organic compounds. Because of the organic compound and other kind of chemical deposition, the center portion of the wood has become very dark in color, too much dark in color. As a result of too dark color wood developed in center, that wood is called heartwood. Sometimes it is also called hardwood, but appropriate meaning is heartwood. But one thing, heartwood cannot transport water. It never transport water. No transportation of water. Sapwood, which is dark in color, can transport water. In this case, heartwood has another name called duramen. Heartwood is also known as duramen. All commercially valuable wood are belongs to heartwood. Sapwood is called alburnum because it is existing in somewhat lighter in color or near to colorless. That's why it is called alburnum. All commercially woods, the rosewood, teak wood, and variety of woods that are available for construction and different types of equipmentation, all woods are belongs to heartwood. Careful, heartwood is not autumn wood. Heartwood sapwood concept is different. Autumn wood and spring wood concept is different. Heartwood and sapwood are found based on physiological maturation. Gradually, when the plant is getting matured, at the time we will get this idea. When a plant is living in a zone where seasonal changes are very sharp, in that part you will develop inside a plant autumn wood and spring wood. Got the idea? This is about the changes that are happening in maturation pattern. Be careful. I have seen many students confuse. Heartwood is nothing but autumn wood. No. Heartwood is a different criteria of development and sapwood is a different criteria of development. This is a mode of maturation or physiological growth. That is a mode of environmental changes. This is about wood. Next. After that, extrastellar secondary growth. Extrastellar secondary growth. When we talk about extrastellar secondary growth, first let us recall what are the changes that we are going to develop here. I am considering this portion is called epidermis. This portion is known as endodermis. This is epidermis. This is endodermis. Could you recall what are the layers of tissues present between epidermis and endodermis? Epidermis, below that hypodermis, below that general cortex, and then endodermis present. All the tissues below the endodermis is called stele, that we have already mentioned. Now I am talking extrastellar area. Now, in the onset of extrastellar secondary growth, there are some cells called cortex cells. The cortex cells will undergo de-differentiation. Cortex cells undergo what, ma? De-differentiation. Then they develop cork cambium. They develop what? Cork cambium. The cortex cells undergo de-differentiation and they develop cork cambium. Now, the cork cambium has a special name called phellogen. Cambium, you are saying it's a meristem, so it has to divide. While it is dividing, it will also produce peripherally, centrally. Now towards the periphery, let me confirm first. Towards the periphery, they will develop some special type of dead cells which are resilient, resistant, and also they are having some water impervious capacity. This is called cork. Name of the cork is also phellum. Phellum is the cork, and it is produced towards the peripheral side. If you talk about cork cells, first of all, they are dead, and they coated by suberin coating. Since they are coated by suberin, they are impervious to water. Water cannot able to penetrate them. Careful ma. Permeable, pervious. Permeable means it will allow, or impermeable means it will not allow. Be careful. Pervious, permeable, these words are closely associated but different. Let's try to understand those words. I am saying something is permeable, that means it will allow them. Okay, done. Something is pervious, something can pass through it. Careful, that word differently. This is a membrane or a layer. I am saying this membrane, if I say this membrane is permeable means the matter what is passing, it would be allowed by the membrane to pass. Okay, done. If I am talking, this is pervious means, look at this thing. Pervious, if I say this matter can pass through it, there is a lot of difference between that. Carefully follow. Both this place, both the area, material passing to the membrane. This is the membrane. Both the places, material passing to the membrane. But there is a lot of difference between permeable and pervious. Permeable means it allowed to pass. Membrane allowed to pass it. Next one. Pervious means this matter has passed. Anyhow passed. But there is a difference. Getting the statement. Impervious means some material cannot able to pass through this membrane. That is called as impervious. Impermeable means this membrane does not allow to pass. Understand? Imagine if I say a membrane is impermeable. A matter will come and hit, does not pass. That means the membrane is impermeable. Got it? Next one. I am saying impervious. Very good. A matter has hit and this matter could not able to pass through it. That is called impervious. Impermeable, I am talking about nature of the membrane. Impervious, I am talking about nature of the matter that is trying to pass through it. Understand? So cortex layer, there, am I clear the word? Should I go one more time? But generally we will talk this word in associated with suberin and cutin layer context, but keep it in mind, impermeable, impervious. Both the time we will talk about a matter passing through a membrane. If you are talking that matter is not passed through membrane with respect to membrane nature, that you will say impermeable. If you are talking this material could not able to pass through the membrane, that is called as impervious. Both are same but different. Then come back to the story. There is a cork. The cork cells are resilient. resilient. If you press them, they will come to original state. And also compressible. They can be compressible. Okay, they can compress them like a sponge. They will come to the original status. That's why this cork is used in cork bend preparation, etc., etc. So this is cork. Towards the inner side, towards the inner side, there will be a development of another kind of parenchymatous layer. This parenchymatous layer is known as secondary cortex. This secondary cortex also possesses another name called phelloderm. Phelloderm. Secondary cortex is also known as phelloderm. Observe. So far, how many layers you have formed from epidermis to endodermis? 3 layers. One is cork. Another one is cork cambium. One more is their secondary cortex, isn't it? Now, all these layers are combinedly has a name called, all these layers, cork, cambium, secondary cortex, all of them collectively considered as periderm. The word periderm. Okay, periderm, secondary cortex, cork cambium, as well as cork cells. Now one more thing. Some areas peripherally, this cork cambium produces some specialized parenchymatous cells. Those parenchymatous cells are known as cork cells. Sorry, they are called complementary cells. Complementary cells. Complementary. Okay, these complementary cells are large and they are parenchymatous cells. They are not going to be compressible. Because of that reason, this area will get blast outside. That means in this area there will be a development of some sort of gap. This gap is known as, this gap will be developed. That gap will allow exchange of gases. This gap will allow exchange of gases. That gap which is developed due to the formation of complementary cells will be in the lens shape. That's why it is called lenticels. What do you call that? Lenticels. Is it okay? Like this, there has been development of peripherally cork cells, below that cortex, secondary cortex, and also cork cambium. All of them called as periderm. Now, one more layer of cell you have to notice, that is secondary phloem. If you come to below endodermis, one layer you will get. That layer has been formed by our vascular cambium. This is vascular cambium. Below the vascular cambium you will find, what do you find? That is secondary xylem. This is secondary xylem. Secondary phloem it is over there. Secondary phloem. Now, the secondary phloem onwards, whatever the tissues present exterior, all those tissues collectively known as bark. What does it call? Bark. Then observe, what do you mean by bark? Bark is a non-technical name. Non-technical name. Bark is a non-technical name over there. What do you mean by non-technical name? Which means this is not technically used but commonly used in representation of different layers of cells, different layers of plant cells. So all tissues exterior to vascular cambium, from here all of them, all are combinedly known as bark. Next one, Bark could be simply denoted as secondary phloem plus peridome. Peridome means all the three layers collectively. Peridome, that's known as bark. Like that, there is a development of various layers of cells, exterior and interior to stele. Intrastellar secondary growth, vascular cambium. Extrastellar secondary growth, cork cambium will be formed. This is about secondary growth in dicot stem. Then go to secondary growth in dicot root. Dicot root. Exactly, the dicot root secondary growth, dicot stem secondary growth, more or less similar, but only one area we will find a slight variation in the mode of operation of the formation of vascular cambium. Vascular cambium will be formed. Just how does vascular cambium form? This mode is mainly differ, remaining all events of secondary growth in dicot stem and dicot root are same. Now I am taking dicotyledonous root. This is dicotyledonous root. Extrastellar area, intrastellar area we will see, but first intrastellar portion directly I am denoting. Just intrastellar area, what changes will be happened, that would be fine for us. Remaining as it is. In this, if you notice, there is a presence of exarch protoxylem conditions in exarch protoxylem conditions when it comes to root. Yes or no? Exarch protoxylem, and also the vascular bundles are radial. On one radius there is a presence of phloem, in another radius there is a presence of xylem. These conditions are called radial vascular bundles. Radial vascular bundle. This is primary phloem, this is primary xylem. The primary xylem, that xylem is called exarch, that xylem is known as exarch protoxylem. Clear? Next one more tissue you have to notice, that is pericycle. Do you remember the importance of the pericycle? These are thick-walled parenchymatous cells in the root, responsible for the formation of lateral roots. So this is pericycle. Pericycle. Next one, when it comes to pericycle formation, it goes like this. After this, see, pericycle responsible for the lateral root. This is primary xylem. In the primary xylem, exarch protoxylem, endarch metaxylem. This is primary phloem. Okay, done. This one I am using to form vascular cambium ring. Why? This is the only event that would be showing some sort of difference in stem and root with respect to secondary growth. What is that? When you talk about vascular cambium ring formation, some tissues of pericycle will undergo dedifferentiation. That pericyclar cells which are present above the protoxylem. The pericyclar cells found above the protoxylem undergo dedifferentiation and become vascular cambium. One more patch will be formed, that is conjunctive cells. I think you know what does it mean by conjunctive cells. The cells which are present between the xylem and phloem in a primary root are known as conjunctive cells. Conjunctive cells will also undergo dedifferentiation. They are also primary tissues. They undergo conjunctive cell dedifferentiation. Then this patch of cambium form, this patch of cambium has formed like that. Gradually these patches will attach. As a result of their attachment, you could see they form a wavy ring of vascular bundle. A wavy ring of vascular bundle. This is first formed vascular bundle is wavy ring. Wavy ring of vascular cambium. The wavy ring of vascular cambium later become continuous ring. Then as usual it started to divide, which means inside secondary xylem, outside secondary phloem. As a result, the primary xylem gradually crush off and remove, and then primary phloem will be removed. Primary xylem is going to crush towards inner side, and the remaining activity is exactly similar to our dicotyledonous stem. But here, vascular cambium, if you see, completely secondary origin. Completely secondary in origin. What do you mean by completely secondary in origin? In earlier, when we talk about vascular cambium of dicotyledonous stem, we said part of vascular cambium is primary, which is intra. Part of the vascular cambium is secondary, that is inter. Here inter not there, completely it is developed secondary. This is the only difference between stem and root with respect to secondary growth. Remaining everything will be same. Okay, this is about secondary growth. As much as I have tried to deliver to you, you follow that much enough, because this part has somehow eliminated, it's still essential for us. That's about secondary growth. Any doubts, you can paste it there. Come on. Do you have any doubts? Any? Somebody saying something? In dicot root will be both primary and secondary crushed. Yes, they will be having same mode of operation, exactly similar to stem. Primary phloem removed first, and then secondary phloem will be removed, and then primary xylem will be removed. Okay. Anything else? That's about today. Happy day. Bye.